For nearly all of human history, our relationship with disease has been a reactive one. We wait for symptoms to show up, for the illness to take hold, and only then do we fight back. Medicine has been the world's best firefighter, rushing in to battle blazes already raging. But what if we could stop being firefighters and become architects? What if, instead of reacting to disease, we could proactively design health, correcting the genetic blueprints of our own bodies before a problem ever arises? This isn't science fiction. This is the promise of personalized gene editing, a technology that is fundamentally shifting medicine from a reactive stance to a profoundly proactive one. We're at the dawn of an age where we can identify the genetic code that predisposes us to an illness and simply fix it. Welcome to G Fun Facts Online, where today we're exploring how scientists are rewriting the code of life itself. The journey to this point has been a long one. Our ability to edit genes didn't just appear overnight. It was built on decades of scientific discovery. The first real attempts at targeted gene editing came in the 1990s with tools called zinc finger nucleases, or ZFNs. Think of these as the first-generation power tools. They were custom-built proteins that could be designed to find and cut a specific DNA sequence. They proved it was possible, but making them was incredibly complex and slow. Next came a tool called TALENs. These were an improvement, a bit easier to design and more flexible, but still a major undertaking for every new genetic target. They were better, but not revolutionary. The revolution arrived in 2012, thanks to the work of Jennifer Doudna and Emmanuel Charpentier. They discovered that a natural immune system found in bacteria called CRISPR-Cas9 could be repurposed into a stunningly simple and efficient gene editing tool. Unlike its predecessors that needed complex, custom-built proteins, CRISPR uses a small, easy-to-program piece of RNA to guide an enzyme to the exact spot in the genome you want to edit. Suddenly, gene editing became faster, cheaper, and accessible to labs all over the world. It was a game-changer that blew the doors wide open for a new era of genetic medicine. So how does this molecular surgery actually work? Let's unpack the toolkit, starting with the star player, CRISPR-Cas9. Imagine CRISPR as a pair of programmable molecular scissors. It has two parts. First, there's the Cas9 enzyme, which is the actual scissor that cuts DNA. Second, there's the guide RNA. Think of this as a biological GPS. It's a small piece of RNA that you program to match a specific DNA sequence in the genome. When you introduce this complex into a cell, the guide RNA leads the Cas9 scissor to that precise location. Once it finds the perfect match, Cas9 makes a clean cut through both strands of the DNA. This double-strand break is the key. It triggers the cell's own natural DNA repair systems. The cell has two main ways to fix the break. The first is a quick and dirty pathway called non-homologous end joining. It basically just glues the two broken ends back together, but it often makes small mistakes, adding or deleting a few DNA letters. Scientists can use this to deliberately break or knock out a faulty gene. The second repair pathway is called homology-directed repair. If you provide the cell with a new piece of DNA as a template, it can use that template to flawlessly repair the break, essentially pasting in a corrected version of the gene. This is how you can fix a genetic typo. But making a double-strand break in our DNA can be risky. It can sometimes lead to unwanted, larger genetic rearrangements. This led scientists to develop even more refined tools. Enter base editing. If CRISPR is molecular scissors, base editing is a molecular pencil with an eraser. It allows for the direct chemical conversion of one DNA base, one letter of the genetic code, into another, all without cutting the DNA double helix. This is huge because many genetic diseases are caused by a single wrong letter in our DNA. Base editors work by fusing a modified, blunted Cas9 protein that can't cut with an enzyme that can chemically change one DNA letter. For example, one type can change a cytosine, a C, into a thymine, a T. Another can change an adenine, an A, into a guanine, a G. By avoiding the dangerous double-strand break, base editing is a much safer and often more efficient way to correct these single-point mutations. But the evolution didn't stop there. The latest and most advanced tool is called prime editing. If CRISPR is scissors and base editing is a pencil, then prime editing is a genetic word processor with a search and replace function. It's incredibly versatile. It can make any of the 12 possible base-to-base changes, and it can also make small insertions and deletions, all without a double-strand break. Prime editing uses a Cas9 enzyme that only nicks one strand of the DNA, fused to another enzyme called a reverse transcriptase. It's guided by a special prime editing guide, RNA, which not only finds the target, but also carries the template for the new, correct genetic information. The system finds its spot, nicks the DNA, and then uses the template to directly rewrite the genetic sequence right there on the spot. It's a remarkable leap in precision and safety, and it could potentially correct up to 89% of all known disease-causing genetic variations. This might sound like futuristic science, but these tools are already being used to cure diseases. The most direct application is for monogenic diseases, disorders caused by a single faulty gene. And we've already had a monumental success story. A treatment called Casgevy is the first ever CRISPR-based therapy to be approved. It treats two devastating inherited blood disorders, sickle cell disease and beta thalassemia. In these conditions, a faulty gene means the body can't make healthy adult hemoglobin. The treatment is amazing. Doctors take a patient's own blood stem cells out of their body. In the lab, they use CRISPR-Cas9 to edit those cells, essentially reactivating a gene for fetal hemoglobin, a healthy version our bodies make as infants. These newly edited, healthy cells are then put back into the patient, where they start producing healthy red blood cells for life. It's a one-time cure, fixing the problem at its genetic source. Similar approaches are being developed for Duchenne muscular dystrophy, cystic fibrosis, and Huntington's disease. Gene editing is also revolutionizing how we fight cancer. The most exciting frontier here is CAR T therapy. Imagine taking a patient's own immune cells, called T cells, and using CRISPR to engineer them into super soldiers. Scientists edit the T cells to express a new receptor on their surface, a chimeric antigen receptor or CHR, that is specifically designed to recognize and bind to the patient's cancer cells. These supercharged T cells are then infused back into the patient, where they act as a living drug, hunting down and destroying the cancer. CRISPR is making this therapy even more powerful by knocking out genes in the T cells that might cause them to get tired, or by making off-the-shelf cells from healthy donors that won't be rejected by the patient. Clinical trials using these CRISPR-edited cells are underway for blood cancers and solid tumors, showing incredible promise. But with this incredible power comes profound ethical questions and significant technical challenges. At the heart of the ethical debate is the line between somatic and germline editing. Somatic editing, which is what all current therapies do, modifies the genes in a person's body cells. The changes affect only that individual and are not passed on to their children. There's a broad consensus that this is an acceptable way to treat disease. Germline editing is different. This would involve changing the genes in sperm, eggs, or embryos. These changes would be permanent and heritable, passed down through all future generations. While it could theoretically wipe out a hereditary disease from a family forever, it also brings up the specter of designer babies and permanent, unforeseen changes to the human gene pool. For now, there is a global agreement that clinical use of germline editing is a red line we should not cross. Then there's the blurry line between therapy and enhancement. Curing a disease is one thing, But what about using gene editing to enhance intelligence or athletic ability? This raises concerns about a slippery slope and creating a society divided not just by wealth, but by genetics, a gen rich and a gen poor. Ensuring these therapies are accessible to everyone, not just the wealthy, is a massive challenge for social justice. And of course, there are the scientific hurdles we still need to clear. Perhaps the biggest is delivery. The gene editing machinery is big, and getting it to the right cells inside the body is incredibly difficult. It's a shipping and handling problem on a molecular scale. Scientists use modified viruses or tiny fat bubbles called lipid nanoparticles to deliver the cargo, but making these systems efficient, specific, and safe is a major area of research. Another huge concern is safety, specifically off-target effects. You need the molecular scissors to be precise. If they cut in the wrong place, they could disrupt a healthy gene or even cause cancer. Researchers are tackling this by designing better guide RNAs and using newer, safer tools like base and prime editing that don't make those risky double-strand cuts. Finally, our own bodies can fight back. The Cas9 protein comes from bacteria, so our immune system can recognize it as a foreign invader and attack the therapy, reducing its effectiveness or causing side effects. So where does this leave us? We are standing at a pivotal moment. The convergence of our ability to read and now rewrite the genetic code is shifting the foundation of medicine. We are moving towards a future where we don't just treat sickness, but we proactively preserve wellness. The path forward is complex, filled with scientific challenges and profound ethical questions that demand a global conversation. But the potential is immeasurable. A future where lifelong genetic diseases can be cured with a single treatment. A future where our own immune systems are the ultimate weapon against cancer. A future where medicine is truly personalized. We are architecting this future right now, one precisely edited gene at a time.